Welcome back to Acing AP Biology. In this lesson, we will be continuing our discussion on regulating gene expression, focusing on eukaryotes. In eukaryotes, gene expression can be regulated at any stage of the process from gene to protein. Some ways that gene expression is regulated in eukaryotes include chromatin structure modification, transcription regulation, and alternative RNA splicing. DNA packing and distance of the gene's promoter from the nucleosome can affect which genes are expressed. In highly condensed chromatin, known as heterochromatin, genes are not expressed. Acetyl groups can be added to histone tails in a process known as histone acetylation. This allows transcription factors to reach genes more efficiently, amplifying expression of those genes. When methyl groups are added to DNA, chromatin is further condensed, making it more difficult for transcription of those genes to take place. For more detail on DNA packing, check out my video earlier in this unit about chromosome packing. Genes in eukaryotes consist of a promoter with a transcription initiation complex that incorporates RNA polymerase II and transcription factors, and then a series of introns and exons. Transcription factors attach to RNA polymerase and each other to help facilitate transcription. Transcription factors bind to control elements, which are non-coding sequences in genes. Proximal control elements are near the promoter and enhancers are further from the promoter. Activators are a type of transcription factor that binds to enhancers. Once activators bind to enhancers, a DNA-bending protein brings activators to the promoter site. Other transcription factors and mediator proteins come together at the promoter and help RNA polymerase II bind to the initiator site. This group of proteins assembled on the promoter is called the transcription initiation complex, and it increases the rate of transcription, thereby enhancing expression of those genes. Transcription factors also include repressors, which block gene expression. Repressors usually act by hindering activators from binding to control elements or affecting chromosome structure. Following transcription, RNA is processed before translation. This processing involves alternative RNA splicing. Alternative splicing of mRNA may lead to the production of different proteins, as regulatory proteins control which sequences of the mRNA molecule are introns and which are exons. Recall that introns are non-coding and exons code for proteins or RNA. During splicing, introns are cut out and exons are spliced together. This mRNA molecule now moves to the ribosome for translation. Therefore, the same mRNA transcript can be expressed in a multitude of ways due to alternative RNA splicing. These are just a few ways that gene expression is regulated in eukaryotes. Keep in mind that gene expression can be modified in any stage of the gene-to-protein process. In the next lesson, we will go over cell differentiation and specialization during development. See you then. For more bio content, hit subscribe.